I can use this to drive another chemical process. But I can't use 7.4 kcals of it, right? I can use something less than that. It depends on how efficient whatever it is I'm driving is in doing it. Honestly, efficiency in biological systems, if you can get to being about 30 to 40% efficient, that's really good. Meaning I'm going to lose about 60 to 70% of that energy as heat, just as waste, but I'm going to be able to use about 30 to 40% of it to do something. And that's what the second law of thermodynamics says, is that I have to lose some of that as heat. We could be a little bit more complex than that, but that's what we're doing. So we're going to release energy through catabolic processes. We're going to use a catabolic process like this in the release of energy as an input to drive some endergonic process. That could be making another compound, building something. That could be pumping ions against their concentration gradient. That could be allowing for myosin heads to swivel back and forth so we can do contraction. Or what? It's all kinds of work, right? And work requires energy, and this is where we get it. But you don't eat ATP, right? You don't eat any ATP. You eat these. And you use those to drive the synthesis of ATP. What part of the cell is going to be really important in understanding the production of ATP?
Mitochondria.
Mitochondria, right? So mitochondria, and the way to say it is this: the enzymes that catalyze the production of ATP, actually as part of something called the electron transport chain, are found in the mitochondria. So that's why the mitochondria produce most of the ATP, is that's where we localize those enzymes. We'll talk about those later. So if you had, here's our kind of structure function kind of thing, right? If you had a cell that was incredibly metabolic, like a neuron or a hepatocyte or something like that, they need a large supply of ATP, so they produce a lot of mitochondria, and they have a lot of mitochondria in them. So we have this ability to specialize. It's also important to understand that we can't really regulate any of these processes without the ability to have information flow in the body. It is important for genetic information to flow. We'll talk a little bit about that later today. Hopefully you're aware that human cells are this type of cells called eukaryotic. Karyo, this root word here, anybody know what that means? Yeah, it means nucleus. Anybody know what this means? True. So these are cells that have a nucleus, right? So a eukaryotic cell is that. Prokaryotes, pro means before, or in front of. Right? So these are cells before the nucleus. So eukaryotic cells have a nucleus, but that's not really, I mean, that is true, but the point of the nucleus is to store the DNA. The DNA is genetic material. It is the information that allows you to produce any protein in your body, as well as many other compounds, RNA, molecules. And hopefully you'll become aware pretty quickly on that proteins in the body provide function. And we have probably more than 100,000 unique proteins that are produced in our body. And that means that we must have at least 100,000 unique pieces of DNA that store this information to produce those. It's a little bit more complex than that, but we have DNA inside the nucleus. The nucleus protects the DNA. Transcription is where we take a nucleotide message of DNA and convert it into a nucleotide message of RNA. And there are some reasons why we have to do that, and we'll chat about that a little bit later. That process is called transcription. RNA then leaves the nucleus, goes out to the cytoplasm where ribosomes are, and ribosomes provide a location where the conversion of information in the form of nucleotides can be translated into the language of amino acids. And a string of amino acids is a polypeptide, which, if it folds the right way, can become a protein. So the goal, the end goal, if you will, of genetic information flow is to take what we've got stored in the DNA and make protein. And if I need a lot of protein, what do? I just do a lot of this. I don't need a lot more DNA. I just need to do this process over and over and over and over and over again. And sometimes that happens nonstop for certain genes. Other times it's regulated. A gene may be expressed only at one brief point in a lifetime, and that's it. It really just depends on what we're looking at.